Hello, everyone, and welcome to my presentation for UCLA's Undergraduate Research Week. My name is Austin Sun, and I'll be presenting my project on the role of cholesterol metabolism in mediating protection against bacterial toxins. I work in the laboratory of Dr. Stephen J. Benzinger in the Department of MIMG here at UCLA. Okay, so first I want to start off by giving some background and also introducing one of the diseases that has motivated this study, and that is necrotizing fasciitis. So this disease is commonly referred to as a flesh-eating disease, and that is because it involves the death of cells within the skin as well as the underlying soft tissue layers. So this disease has a very high mortality rate, and also one of the main issues is that there are currently no preventative treatment options, but hopefully the information uncovered in this study can help progress towards that in the future. All right, so necrotizing fasciitis is caused by certain gram-positive bacteria, and one of the toxins that is secreted by these bacteria is the cholesterol-dependent cytolysin, also known as the CDC. The CDC will be the focus of my study, and it is what contributes to the degradation of the skin that you saw in the previous slide. Okay, so a little bit about the structures of a CDC. So they are composed of 4 different subunits. More specifically, subunit D4 is responsible for recognizing and binding to cholesterol within the plasma membrane, and subunit D3 is responsible for penetrating the plasma membrane. So these CDCs, they require the presence of cholesterol in the membrane to function, and their main function is to form a pore within the cell membrane and therefore lead to cell death. So here's a general schematic of how the CDCs function. So first, they will recognize the cholesterol within the membrane, and then they will penetrate the membrane. Now, CDCs, they mostly exist as monomers. So once they have been bound, they will recruit other monomers to form an oligomer. Now, once this oligomer has been formed, they will create this pore that spans the entire cell membrane. Now, this is very lethal for the cells because it disrupts the cell's ability to regulate its own environment and also protect itself from anything in the extracellular environment. So the result here is almost always cell death. Okay, so the primary focus of this entire study is cholesterol. Cholesterol normally is very important for cell function because it maintains the structure of the cell membrane. Cholesterol is also the most abundant lipid in the human lipidome, composing 30% of all lipids. And most importantly, as mentioned earlier, the cholesterol is essential for the CDCs to function. So the idea here is that perhaps if we can sort of modulate the amount of cholesterol within the cell, that can give an individual protection against the CDCs and therefore decrease the effects of diseases such as necrotizing fasciitis. Okay, so on the topic of cholesterol metabolisms, one of the main pathways that has been heavily studied in previous literature is the liver X receptor, also called the LXR. So LXR is a transcriptional factor, and in its basal state, it exists as in a repressed state, so with the co-repressors bound to it. But once you add a ligand to it, the co-repressors are replaced for coactivators, and this leads to the expression of multiple genes, such as ABCA1, and the function of these genes is cholesterol efflux. So that basically decreases the amount of cholesterol that is present within the cells. All right, and then the subject for this study is macrophages, and we chose to use macrophages for multiple reasons. First is that they have very high plasticity, which means that they respond quickly and efficiently to environmental stimuli. They are also relatively easy to culture and expand, so that gives us a lot of cells to work with. And they are also present throughout the entire body with a very diverse set of functions. And then they are also rich in cholesterol, which is great for this study because we are focusing on cholesterol metabolism. Okay, so now hopping into experimental design. So first we start off by isolating the femur and tibia bones surgically from a mouse. And then we flush out all the bone marrow within the femur and tibia bones. And then we culture these cells in a specific medium that allows us to differentiate them into macrophages. So we're generating bone marrow-derived macrophages, also known as BMDMs. So we let the BMDMs grow for about 3 days, and then we'll change to fresh media. And usually by day 7, they're fully formed and matured. So at that point, they're ready for any sort of treatments or stimulations that you want to give them. All right, so once we have our bone marrow-derived macrophages, we then stimulate them with an LXR agonist. In this case, we are using a synthetic ligand called GW3965 for about 24 hours. And the purpose of this is to upregulate the LXR pathway and increase the expression of those cholesterol efflux genes. Our next step here is to stain it with two different dyes. So we use Host as well as propydium iodide, also known as PI. Host marks the nuclei of all cells within the sample, both alive and dead. So that gives us a good baseline to compare to. And then PI specifically only marks dead cells. So in the context of this study, if a cell is infected with the CDC, that creates these pores on the plasma membrane. And only then is the PI able to permeate through the cells and create that signal. Without the pores on the membrane, PI wouldn't be able to permeate, and therefore there would be no signal. And the purpose of PI would just be to mark dead cells in this experiment. And then at this point, we challenge the cells with a CDC. In this case, we're using a CDC called SLO. And then after that, we perform live cell imaging, and we're measuring the amount of PI signal within these cells. And we're comparing that to our baseline to see how much cell death is occurring once you add the CDC. And then finally, we will graph our results and compare the different treatments. So here's an example of an image from live cell imaging. Here in the left column, we have no CDC added. And what you'll note is that for propydium iodide, there's a very low amount of signal, which is what we expect because we're starting off with cells that are all alive. And therefore, because PI only marks cells that are dead, we don't really expect to see any or very much signal at all. Now, moving over to the right column, when you add CDCs for about 20 minutes, you'll see that there's a very significant increase in the PI signal. And this is to be expected because once you add the CDC, that creates these pores within the cells, the PI permeates through and causes cell death. And therefore, we're able to see more PI signal. And then here at the bottom, we have Host as a baseline. So because Host marks both dead and alive nuclei, we don't see any change in these images. And that is to be expected. And this gives us a good baseline to compare. All right, so here are our results on a graph. The first treatment here is the blue line, which is no SLO, and this is just our negative control. And then looking at the red and green lines, so this is in the wild type mice, and we're comparing two treatments. DMSO is our negative control, and GW3965 is our LXR agonist. So this is specifically upregulating the LXR pathway. So comparing these two lines, we'll see that for the green line, there is a very low amount of cell death, which is shown on this y-axis here. The y-axis is the number of PI positive cells, which essentially means the amount of dead cells. So looking at this green line, there's a very, very low amount of cell death, very comparable to the no SLO treatment. Whereas if we look at the red line, we see that there's a much higher percentage of PI positive cells, which means there's a larger proportion of cell death. This is interesting because it shows us that potentially by stimulating the LXR pathway, there's some sort of protection going on that is protecting the cells from the CDCs. And then finally, looking at the purple and orange lines. So these two treatments are when we knock out LXR in the mouse. And what we see here is that for both of these, there's a very high amount of cell death. So that kind of shows us that LXR is pretty important for this protection against CDCs. And once you knock it out, there's a very high amount of cell death, regardless if you stimulate it or if you don't. Okay, so for our conclusions, our graph has shown us that the presence and stimulation of the LXR pathway seems to protect the cells from the CDCs. And then this effect is also specific to the LXR pathway because, as shown in the graph, once you knock out LXR, there seems to be a high level of cell death regardless of what treatment is given to it. Okay, so for some acknowledgements, I'd like to thank Dr. Benzinger, my PI, as well as Min Sub Lee, who is my mentor. And then, of course, thank you to the NIH for funding this project. All right, that is everything. Thank you so much for listening.